Great. Well, thanks very much, everybody, for coming back. It's wonderful to see so many faces in the audience, and it gives me great pleasure to present John Cleary to you. Now, John is a university lecturer, and he is the project coordinator of Cromwell's Scottish Prisoner Tracing Project, among other things. He lectures and teaches at a university in Edinburgh. He's a member of ISOGG, the International Society of Genetic Genealogy in Scotland, and is involved in a project researching the fate of Scottish prisoners captured by Cromwell in the Civil War and transported to the Americas using DNA and genealogy, working with the prisoners' descendants. Now, John is going to take us through next generation sequencing today with tests like the Big Y and various other tests that you can get from family tree DNA. And he is going to tell us how these type of tests have made huge advances in genetic genealogy in the last couple of months. And he'll also be talking about Y-STR markers and how the two correspond with each other. So please give a big welcome for John Cleary.
Thank you, Maurice, and thank you everybody for coming here today on this very beautiful afternoon. It's a shame not to be outside. Now the sun's shining finally, but if I speak quickly, we can get out quicker than I finish. Okay, so I'm going to talk to you, as Maurice said today, about Y chromosome testing, in particular next generation sequencing. And can I just ask you, have any of you either taken the Big Y test, or do you run a kit for somebody who has taken the Big Y test? A few of you. Are any of you thinking about taking the Big Y test? So there's some interest here. I'm just maybe just curious to hear what it's all about. Right. I'm afraid I'm going to assume that you're all familiar with the basics of Y chromosome testing. So to save time, because there'll be another talk to the topic, I'm going to presume that you do know what STR testing is, and you're familiar at least with that particular approach to working out Y chromosome lineages. So I'm going to start by looking at a few very common ideas or notions I've been expressed about next generation testing, which no one's actually said these things, but they kind of sum up things people have said to me. Such things as it's all about deep ancestry, it's all about finding our cave person ancestors, that knowing what your haplogroup is might be very nice, but won't actually help you with genealogical research. And that STRs are enough, and we don't need to move into the zone SNPs. Now, I think all of these are wrong, and I'm going to try and explain why I think they're wrong in the course of this talk. I'm going to talk about the types of next generation sequencing tests which are available to genetic genealogists, and I'll look at how we can then use the results, because they do generate a huge amount of data. And one of the biggest problems that any of us may face is what to do with all the data that it generates. So I argue that subclades and haplogroups and SNPs are becoming very important now in helping to identify people who may share a common ancestor with us, but also those who do not. And I also believe that in the current period, the current year, we're now seeing SNP testing becoming increasingly relevant for historical family research. In other words, the markers which have been discovered are helping to identify branching taking place in the historical period, or what's often known as the period of surname use. In other words, the time that you are probably researching as genealogists, when there are paper documents which you can correlate with your Y tests. So that's the wrong title. It should be SNPs. What are SNPs? First of all, very quickly, it stands for single nucleotide polymorphism, but more simply, these are point mutations at a single point in the Y chromosome chain. So here, for example, is a little snippet from a diagram of part of the Y chromosome. Down here we have the Y chromosome reference sequence, and here underneath these blocks we see a C, and when a SNP occurs, the C may change, for example, to a G, and then you have a change sequence in the Y chromosome. And people who have this particular mutation, it's a real mutation, carry a marker known as L21, and I bet that most of the men sitting in front of me will have this marker if you're from the British Isles. I do, many of you will have it too. So that's what a SNP is, and of course these markers like L21 have been known for a very long time, and they've been seen as indicative of deep ancestry. L21 appeared in one man about 6,000 years ago, roughly, give or take, a few thousand years, and all of us therefore who carry this marker are descended from that one man. He is a common ancestor. All of us have, how many generations back? 150? Whatever. A long way. But of course there are SNPs occurring all the time, right the way down from this person before and upwards to the origin of the human race, and of course from this person downwards, and SNPs are being discovered which may have occurred in your father, your grandfather, great-grandfather. Such SNPs are all now being discovered in people doing these kinds of tests. So one of the great dreams of many genetic genealogists is to find the SNP that will predict their surname. And I've heard people speculate that this may be something on the cards in round about 2017 or so, once we have a lot more tests like this and once a lot more definition begins to appear in the trees. There are some possibly predictive SNPs appearing already which seem to be associated with particular surnames. I mean, careful how I phrase this, but I've been in touch with a couple of project administrators about whether they think this is the case for them. I was in touch with the administrator of the McFarlane project, who believes that these two numbers here, BY674, 675, BY means they were discovered in the Big Y test, are indicative of McFarlane. So if somebody was to test and be found to have these SNPs, then that man would be either a McFarlane or somebody descended in recent times from someone named McFarlane. That's what he believes anyway, and I think he's got some very solid grounds to believe that. He told me that there are a few surnames down here which are not McFarlane. For example, there is Irvin over here, and he believes this is a recent NPE within this particular McFarlane branch. And so there's one more down here, McAfee. I don't know how this fits in, but in general, there seems to be a strong correlation so far between these surnames and these particular SNPs. Now, we must be careful. Of course, we do not know how this sample has been constructed. No doubt the McFarlane project is recruiting McFarlanes, maybe with a certain known relationship, and therefore this is not a genuine random sample. And so there could be other surnames we do not know yet who may be associated with this. But at the moment, it looks quite encouraging for McFarlanes that they are developing distinction in the tree with SNPs probably occurring about 300 or 400 years ago, which may predict people to carry that surname. There's one more which is in the family Maxwell. Again, I've been in touch with a particular project administrator. He's a bit more cautious. He points out that, again, there are other surnames like Leman appearing here. But again, we seem to have this block at least, seems to be predicting, again, the surname Maxwell, or the particular branch of Maxwell at least. But there's another problem here we must be very, very careful of, because the Maxwell administrator believes that all of these SNPs above, and it goes way up, there's a lot more I haven't put in my diagram, are also going to be Maxwell SNPs. But these mutations probably appear in people every 150 or so years, give or take, again, a few tens of years. And so these probably stretch right back to way, way, way before the beginning of surnames. So some of these may indeed be Maxwell SNPs, some of them will not be, and will be shared eventually with people with other surnames. The problem is which ones are which, and that of course is one of the big challenges to anybody who's doing this kind of testing, because you get long chains of mutations like this, and we've got to somehow begin to sort them out, try and get some hierarchy into the structure, and work out what actually are the genuine branching patterns, and how far can these be correlated to particular surnames. However, these are two surname projects, branches of surname projects with genetic families sharing a surname, sharing some distinctive SNPs, and again, they've made this big advance in the past 12 months. So that's the kind of dream that is moving many people on with this kind of research. What actually is next generation testing? Well, it's next generation sequencing rather. It is a form of sequencing stretches of your genome, not necessarily the Y chromosome, any part of your genome, and essentially it is the big advantage of this is that it is fast and cheap, or faster and cheaper compared with previous technologies. And this is made possible for genetic genealogists like yourselves to have access to relatively inexpensive tests which will use this particular technology. What it does essentially in brief is it smashes up your stretch of genome into its little bits. Each one is about 100 base pairs in length, but the important thing is that they are overlapping. So it must break up your stretch of the genome at random places, so you get lots of little stretches which can be easily read, and they overlap with each other. And then very clever software can then begin to build up a picture of the whole genome by fitting these pieces together like a jigsaw. And this can be done by computers in a few hours once the data is available. So this is what enables these tests to be done very quickly and, again, in relative terms, quite cheaply, and they're getting cheaper. So big NGS tests available to the genealogist at the moment come in two flavours from two companies, one of which is our friends across the walkway there. This is the big Y test, and the other is run by a company known as YFull Genomes company, and they have two versions of their test, although from what I gather from their website, these now seem to have merged into one. I'm not quite sure, but I think there's just one test there now. You'll notice that there's a big difference between them in one sense, in that the big Y test is testing fewer positions than the FGC tests, but I would argue that there is a very important cost-benefit balance here. If you test fewer positions, the test is cheaper. And I think there are strong reasons to go for a smaller test, because you'll get enough information to handle, and because it's cheaper, you'll be able to have more of them and potentially get a comparison between two people. So I rather regard the FGC test as being more the purist, who wants to know everything about their Y chromosome, whereas the big Y test is a bit more pragmatic. It also, I think, I understand, tends to have slightly longer lengths of the Y chromosome read as well, which may be an advantage in identifying certain SNPs. So we talk about the readable Y chromosome, a big Y covering about 55% of this, the YFull Genomes test approaching 100%. What do we mean by the readable Y chromosome? Well, I'll just quickly whiz through a bit of did you know stuff about the Y chromosome in case you didn't know. I didn't, so I began researching this. And in fact, about half of the Y chromosome cannot be sequenced at all with current technology. So we're only really looking at about half the chromosome when we do these tests. Some bits of it recombine with the X chromosome, and therefore they're not passed on intact from father to son. And then about half of the Y chromosome is this region which is repetitive, and therefore cannot be easily sequenced, and is therefore beyond the reach of the current NGS test that we would use. So we're really looking at this much smaller chunk here, which I think is itself only about 45% or so of the full length of the Y chromosome. Let's look at this in more detail. This also is divided into a number of regions, some of which are probably better than others for doing the test on. We look very closely, very quickly at these. We refer to the X-degenerate region. Since it is the Y chromosome, it's degenerate apparently. The ampliconic region, and there is the X-transposed region. Quick quiz question. Which of these three regions do you think is the most useful, potentially, for this kind of testing? Degenerate, transposed, or ampliconic? It is, of course, the X-degenerate, which appears to be the richest in known SNPs and potentially in other stable SNPs which are useful for the kind of branching work that we want to do. Essentially, it's degenerate in the sense it was originally an X chromosome, which then degenerated into the mammalian Y chromosome. So you could really call it the X-evolved chromosome rather than degenerate. It sounds better, I think. The other two, there are parts of the ampliconic sections which contain the dreaded palindromes, and again are rather repetitive, and therefore SNPs found in those regions may be less reliable or less robust. And then the X-transposed section is a big section, the purple bits, no, the red bits of the Y chromosome, which were transferred over from the X chromosome at some stage in our evolutionary history, but resemble other chromosomes very, very closely. So when you do this kind of testing, you can't be sure that you're finding something on the Y chromosome or on one of the others. So I think that a strategy of developing a test for genealogists, which doesn't aim to cover all of this, but just essentially targets the parts that may be most useful, and in consequence produces a cheaper test with maybe a slightly smaller amount of data, is actually a very good strategy. And therefore, I think that the big Y is great value for genealogists to consider. So I think it gets the affordability and coverage balance about right. It is. It also means, because of the higher volume of big Y testing done, there have been about 800 in my L21 haplogroup alone in the past 18 months, and many more across other haplogroups. So the numbers are growing considerably. And it does seem to target areas where known SNPs exist that can be used for identifying haplogroups reliably, but also which may be rich in potentially stable SNPs to identify new branching and new subclades below them. Now, some people sometimes ask about some of the fixed SNP tests. Some of you may have done Geno2. Has anyone here done the Geno2 test? A few of you have, or the Chromo2 test. Yep. And again, what these do is they have a fixed menu of SNPs which they test for, but they're not discovering new SNPs. So what the big Y and next generation sequencing can offer is discovery of unknown markers. And if you want to try and find out what may characterize your family branch in terms of markers, you may need to discover new ones that will be particular to that branch. These fixed SNP tests cannot do that, but the big Y can, and that's why, again, I think is of great value for genealogists. So, I'm going to go very quickly through this, but you have a chance to ask me questions at the end. So, if you've done these tests, you'll be aware that there is a huge volume of data generated by them. Some people refer to it as the data tsunami, and there's various strategies that have been developed in the past 18 months for trying to handle this. And to be honest, when I first saw my results, I thought, what am I going to do with these? And you may feel the same if you've done this test. So there are a number of things you can do, one of which is go to the large haplogroup projects. If you know your haplogroup, if the project is active, there will be experts there who will be able to advise you and maybe process your data for you and help you see the patterns and the structure in that data. So I've got a few examples here. Since I'm in haplogroup R, I know more about some of the big sub-haplogroups within R, and I know that these four in particular have got very active administrators who are developing dynamic trees for their group. When I say dynamic, I mean that they are updating them regularly as more tests are done, as new data comes in. They're building more structure, more refinement into their trees. They're very active. I'm going to show you some examples from one of these, which is the P312 big tree, been developed by a man called Alex Williamson, a very dedicated man, who I think in some ways is coming with one of the very best tools for all genealogists to use. So, Alex Williams' big tree then, as it says here, is using NGS data from the two tests, but not from the old fixed SNP tests. They're not useful here. The are discovery tests, and he's trying to reveal the novel SNPs, the new SNPs. Indels, that's a term for things that are inserted into your DNA or deleted out of it, which are also kinds of SNP mutations, which may mark new branches. This is the key thing, of course, for genealogists, finding where a block will divide and two new branches are created. And this lends itself to the tree structure. And here, for example, is the top of Alex Williamson's tree, showing P312, the original granddaddy of them all. Up here, obviously, there's a lot more above him, of course, but he's focusing on P312. And then these are the major branches so far known, P312. And as I said, a lot of you, including me, would be in L21 and below. So that's on the top level. L21 itself has, below that is DF13, has about 20 odd branches. So we're talking about an enormous tree. It's getting wider and wider and can't exist in a single page. So here is my own particular subclade, which is known by the awful, dreadful name of FGC 5494. One thing to get used to when you're working with these SNPs is the dreadful labels they use. I try and see them as catalogue numbers. If you have a good index, you will look up where they are, which haplogroup they're in, and so on. You don't need to remember any of them unless, of course, they're relevant to your research. So these then are all the various branches of this branch of a branch of a branch. This was recently discovered itself, only about a year and a half ago, I think. And down here you'll see the Maxwells, as I was mentioning earlier, so they're part of this group here, that long chain of SNPs they have here. This top one is probably about 4,000 years old. So we're talking again a long time depth, but it does go down to the present day. So all of these people down here are people who've done tests, and therefore they are living today. And in between them and here are the various SNPs which have been discovered in the tests they've done. Over here in the far end, you'll see a long chain with no SNPs on, and some fellow called Cleary down the bottom, who doesn't seem to have any SNPs. I do get this last of my tribe feeling sometimes, but there's a reason for it. I just don't have any matches who've done Big Y yet. But one of the other things that Alex is doing is essentially, these are all shared SNPs. These are all shared by at least 2 people. And of course, I have a lot of mutations in my line, which I share with nobody yet. Please test over there. Maybe one of you might share with me. But anyway, I do believe that somebody will pop up sharing some of these, and then I too will begin to get a bit more definition in my tree. But these are all the private SNPs that Alex Williamson has identified in my raw data as being relevant to me, and he even classifies them according to how good or how valuable he thinks they are. I won't go into that now, it's quite complicated, but it's good to know that this has been done. And one of the other things that you'll be able to get from NGS tests, by the way, is more STRs than you may have previously had. And Alex Williamson is trying to build STR data into his analysis as well by looking at the haplotype progression from the top down through the subclade to the tester, and seeing, therefore, which SNPs may have mutated, sorry, which STRs may have mutated in which order. Obviously, there's not very much here because I haven't got any matches, but if I did, then there could be several more layers here showing some ordering in which the STRs themselves may have mutated. So again, people like this are doing great service, I think, for people like yourselves who may test and not be quite sure what to do with the data. There are third-party analysts. These, of course, are commercial concerns who will charge you a bit extra money for the service they provide. And I've done it myself. I've gone to one of these. I think it was worth it. But that's a decision that people must make. However, they tend to charge a fairly relatively small fee, about 10% of the cost of the original test. And what you'll get in many cases is, again, more structure and understanding of your tree. This is, I'll go through a few of these very quickly. We've got Clarify DNA are building a dynamic tree based on raw data. And if anyone wants to look at the web addresses later, they're all on the slides down here. The Full Genomes company will analyse raw data from other companies as well as test their own. Though again, what you tend to get from them will be a big zip file full of spreadsheets. It's not necessarily the easiest thing to navigate through if you're not comfortable using spreadsheets. You can also view your own BAM file. This isn't a third-party analyst, but you can view your own raw data, it's called a BAM file, through some viewers such as this one called IBG. And here, for example, in this little window, I've taken a relative's results, and these lines are all the separate reads. In other words, these are all stretches of 100 base pairs that were read by the test. And you see, they don't line up, because the aim is they overlap with each other, not line up. And therefore, some of them have this particular point, and some don't. Down here, you see the reference sequence result was a T. In my relative, every read is giving an A. So we can be pretty confident that there is a good mutation here, a good new SNP, and there is. We call this YP355. This was discovered just exactly a year ago. And in fact, there's a lot more. It's actually better than this. It goes all the way down. There's a lot more reads than they're showing if you zoom out. So you can do this yourself, but again, there's a bit of a learning curve in getting to be familiar with this. One of my own favourite third-party analysts is one that offers, I think, a lot more service for genealogists. And this is, I think, what genealogists who are not necessarily trained how to use data would like to have, would be a graphical or visual presentation of results that will allow them to go into their raw data, their BAM file, and to navigate it easily, to find out what's in there, what's interesting, and to work out a way of evaluating the calls, the new SNPs which your test has found for you, and also provide you with a dynamic tree that will be updated as new data comes in. And I think because genealogists too are concerned with time, you're looking at periods when surnames may have been used or when there are paper documents to correlate against your genealogy. So you'll probably be interested in knowing if a branch is likely to have occurred, say, within the last 400 years or 800 years, whatever. So dating is a difficult thing, and we have to be very, very careful with any estimates of dates of mutations, but it's something which you would be interested in. And there's one company which is trying to do this, which is YFull. And YFull they only analyse raw data files. If you test, you can send your BAM file to them, and in about 5 weeks they will analyse it for you and produce a report with lots of nice visual graphics showing you your known SNPs. So these are SNPs that you have that are already known. They'll show you STRs, more STRs than you may have tested already. YFull are reporting about 440 STRs, the most. There's 490, but nobody has all of them. And it's possible to compare these results with the family tree DNA panels, but always take the family tree DNA panel as being more correct than this if there's a discrepancy, because this is still very much in its infancy. But it's there, and it's something which could become more useful, more important in the future as the technology is refined. And of course, very importantly, they will tell you your novel SNPs. This is what you really want to know. What are the ones you have that have not been found before, that may be yours or may be shared between you and members of your family or more distant relatives? And so YFull will present these. This person only has one, I50, but don't worry about that. That's because this person has a lot of matches, and therefore the branching starts much more recently in time. And YFull offer a very dynamic tree. They update this weekly. As new tests are analysed, they update their tree. Now, again, it's better for some haplogroups than others. That's the way the cookie crumbles, because some haplogroup administrators are more active in forwarding their testers to YFull than others. But again, you will find that this is a very dynamic resource, and you've got to find yourself in a tree. It's public, by the way, but identified only by YFull number, so that preserves privacy. But if you know your number and the number of your matches, you've got to find your location on their tree. So, I think that YFull offer 3 particular things that will be of great use to genealogists who want to use this kind of testing for their projects and project administrators. I think 3 things that we will really, really want to know about this kind of data is, we want to be able to look at anywhere in the BAM file. In other words, the BAM file is your raw data, and I've shown one graphical view already, but I think any analyst, any analytical report will need to give you a means to inspect any part of your raw data. If you find a SNP there, you'll want some means to evaluate how good, how reliable, or how unreliable that SNP may be. And you'll need to check all of your new SNPs for what we call phylogenetic consistency. Put a mouthful there, but this means, is it something that's consistent with a tree? Can this SNP be used to identify a branch? Or is something which seems to be sprayed around and doesn't have any consistent pattern? There are a lot of false positives in these tests. I don't know what proportion of calls will be false positives, but you must be suspicious of some reads, and therefore you need to check any new SNP you find to make sure it really is something that's occurring in one place, or one branch, or one block. And therefore, YFull, I believe, offer a means to do all 3 of these things. First of all, I have a BAM file viewer. I'll whiz through here. Let's check, obviously I don't have a live connection here, but let's check the position. Here's one I prepared earlier. This is a position on the Y chromosome, and we can search for this, and YFull will then give the data, some summarised data from the BAM file. Very interesting here, we see that it's been read 71 times in the process. That's very good. Many positions are read only once or twice or 3 times. They're obviously much more suspicious. 71 times is pretty good. And every single read has come up with an A. And we see down here that T is the original, A is the mutated form. So in other words, this result is definitely the mutation. And this is actually the same one I showed you earlier on the IGV graphical viewer. This is the one we call YP355. You'll hear more about that later on. So that's good. You can also look at how good these SNPs are. The one I showed you there, by the way, had a 5-star rating from YFull, and a few of them are discussing how YFull derive their ratings. I'm not entirely sure it's totally transparent, but I think it's a reasonable guide at least, or I'd like to know more about how they do it. But you can use this to evaluate any new call you have. So let's, here's a SNP that was called recently by somebody in my group. They called it YP1418. I was actually suspicious of it from the start, and so I searched for it in one of my members to see if he had it, and he did. He's believed positive for it, only 1 star though, so also looking a bit suspicious. And YFull, a very neat little function. This little magnifying glass next to the read allows you to click on there and get the data from the bound file. And here it is, and this one is quite worrying. This is not a SNP I put any faith in. We see here that there are 3 different results. Down from here, the original is C, the mutation is G. So 38 Gs out of 48, that's not such a good proportion. 7 are on the original, and 3 suggests there's nothing there at all, it's been deleted. So something inconsistent here about the result. And YFull give us some metrics, and this is a very important one here. I think I've got to use my yes clicker here. So only 79% are read with a mutation. I think 90% is a good guideline threshold. And YFull have this probability of error. Essentially they're saying there's a 26% chance this is wrong. In other words, only 74% reliability here, and as I said, we need the 90 ideally. So I would actually reject this as being a SNP of interest. I'd discard it from any tree building I was doing. So the last thing I mentioned is the check of phylogenetic consistency, and YFull have a very neat function here, which are groups. These are very similar to the haplogroup projects you may have joined if you're a family tree DNA tester member. And there's one here for R1A. These results are in haplogroup R1A, and all of the YFull testers have joined this one group. That's a very, very useful function because it means that we can test all of them for any new SNP we find. We can search for that, see what the result is for that SNP. So I can type in the position for YP355, do a quick search, and there's actually a great long chain of these 300 people, and the only ones who are positive is this block of A's here in the middle. And that's good. It's telling me here this block represents a branch of some kind, and it does. And we're pretty sure that YP355 is a very interesting new branch marker, which some of us have been studying for the past few months. On the other hand, you may get a result a bit like this. This is an unnamed variant that cropped in one of my group members, and this is not going to be useful. Quite from a visual scan here, there's no consistent result. There's some T's, some A's, spread all over the place, and with these strange W's, you may know that the base letters for the bases of DNA are A, G, C, and T. So W is not one of them, but this means unclear result. 4 A's, 4 T's, not clear which one it is. So this really is not really useful. And again, we disregard that as being not useful. So all of these are very useful for people researching their own genealogy or project administrators are looking for a means to make sense of the data which you're generating. You can also analyse your data yourself. Don't be afraid. I've been working with the joint administrator of the Cummings surname project, which we administer together, and we have developed our own quick and dirty system for identifying new SNPs and people in our group. And we actually take our data from Family Tree DNA's results page. Many people have suggested that it's not very refined, and that's certainly true. You could do with some refinement, but all the information you need is there. And if you tested Big Y, you have a page showing all of your novel variants, which are your new markers, not shared with somebody else, according to Family Tree DNA. And you can either find them there, or if you want to work on someone who matches you, then you can go to your Big Y matches page, and you'll find the same data in 2 windows, all the SNPs they share with you, and all the unique SNPs he doesn't share with you. And again, you can extract these into a spreadsheet to work with them. So the problem with Family Tree DNA's results list is everything is there, but there's too much there. And actually many of these novel variants are not novel at all, they're known, and many of them are actually already established on the tree. So we need to filter out the ones which are not necessarily really new variants, and so Tim Cummings, the joint administrator of the Cummings project, has developed a very neat filtering algorithm in a spreadsheet to do that exactly. First we put all of these new positions in, they're all marked by position numbers for the Y chromosome, and first we filter out the ones which are known good SNPs in the tree, get those out of the way. We then filter out all the ones which are rubbish, potentially, or we think are not going to be reliable, robust markers, get them out of the way, and this leaves us with a shorter list of genuinely new markers that have not been encountered yet, which may be anything from 1 up to 40 or 50, depending on the tester and the branch. And then these can be filtered a third time to see if we've found them already in our group. We're very interested in this, because of course we're looking for those markers which may mark branches, which therefore may be in more than one person's test results. And we found a few of these, because this one is cheating slightly, because I'm actually filtering here against the same person, so obviously they all match. But we may find in doing this that one or two will come up as being known already in one other person. And because we now have it in two people, we have the potential here to investigate this new position as a new SNP, as a new branch marker. Of course, there's more to it than this, we're going to check it for robustness and so on. But this is how we find ones that may mark new stages on the tree, and we're not building our own tree. So I hope I'm not really out of time yet. Morris will wave at me if I am. Okay. I'm going to try and illustrate what we're doing by looking at a case study of Kemp and Cummings. So Kemp is my maternal line, my mother's name is Kemp. My grandfather was from County Cavan in Ireland, and I've been investigating her line through a DNA study with my uncle, who's very enthusiastic about this. And I began looking at Kemp's in Ireland about 3 years ago, genealogically. And Kemp's a very interesting name in Ireland. It's very quite common in England and in Scotland, and it's both seen as an English and Scottish name. But in Ireland in 1901, there were about 200 people only with the surname Kemp. Many of them were English or Scottish blow-ins, so actually it's quite a rare name in Ireland generally. And there are 3 pockets of Kemp, in County Cavan, where my grandfather was from, in Cork, and in Limerick. I know nothing about them yet, but obviously when I began doing my genealogy research, I had a very obvious initial hypothesis, or question, which is, were all the Kemp families in Ireland related to each other? So I set up a hypothesis, which is to say, yes they are. I can then test that hypothesis by doing some STR tests on a selected number of Kemps. The Cork Kemps very kindly done one a few years earlier. They were there to be compared with. I added a few Cavan Kemps, and immediately the answer is no, they're not, because they're totally different haplogroups. The Cork Kemps are R1b, U106, and the Cavan Kemps mysteriously came out as R1a, which is also a fairly rare haplogroup in Ireland. So, my next question then is, well, are all the Cavan Kemps then related to each other? And we're going quite quickly through this. There are as many as 26 families in County Cavan in the 19th century, using the name Camp or Kemp, which are variants and interchangeable with each other. There is only one left now, by the way, quite sadly. They've all gone bar one, but they believe they're not related to each other. There's no evidence of relationship, and so Kemps in or from Cavan would say, yes, that's your family, this is mine, we're not related. So that was my hypothesis then. These, doing the other way around this time, these Cavan Kemps are not related to each other. Can I falsify, disprove that hypothesis? I wouldn't claim proof as such, but the STR results show very strong evidence of relatedness amongst all these families. I was able to test 8 of 9 lines that I identified, and eventually I coalesced these. These are the 9 lines. The oldest one in the middle, I wasn't able to track this down. I think they've gone. But I was able to draw probable links, the red lines between some of them, and possible links between the others. And I think that there are now 3 broad lines which coalesce into a single common ancestor sometime before 1700. When before 1700, I can't say, but before then. Now, this is a tree I've built using the STR data only. So as a check to make sure I'm not falsifying or looking for, or using confirmation bias to look for evidence of relationships. I want to see it. I'm trying to see whether the STR data I had on 6-7 markers could be built back into a logical tree with logical positions for common ancestors at each level, and it can. And I can link it back to something called JAX. I'll tell you that in a moment. And the only thing about this tree is there's no time depth on this. This could be 300 years or it could be 3,000. The point is that there are common ancestors at these branching points, and these branching points are identified by STRs. What I want to be able to do is try and see whether I could use SNPs to refine these further, but also do something else onto the JAX. I began to notice that the Kemps in Cavan had a lot of matches who were not called Kemp. And of course, many of you have had this, and a lot of people say disregard them. They may be NPEs, you may not be able to prove a relationship, they may be false positives. All of these things are potentially true. But I began to ask myself, are these other people actually related in some way? I know that the Jacobs family have got no evidence of ever having been Irish, but they know who their common ancestor is, and that puts them in a strong position in one sense. This was a man named John Jacob, who went to Virginia in the 1660s as an indentured labourer, and all the Jacobs in that part of America are descended from him. They've done a major STR test, and they've demonstrated their relationship. So we have that to benchmark on here, 1660 as a time when the Jacobs coalesce. And then there's other names such as Cummings, Anderson, and I've been in touch with some of these, and I kind of began to wonder, well, all mixed together. Is there a surname cluster here? Is there actually something interesting I can explore here? So this is where we bring in the Jacks, and I say this quite safely here because I used this talk, probably this talk in Dublin last year, and Jacks has other connotations in Ireland I won't go into just now. It wasn't me who came up with this acronym either, but it does summarise the major names which we're looking at, and in particular we've been looking at Jacobs, Cummings and Kemp in STR testing, and Cummings and Kemp have been doing all the big Y testing to see how this may stretch. So we know that these have some distinctive STR markers. This one here has been called the so-called young Scandinavian marker. I mean, I'm trying to get away from using terms like that, but it definitely seems to be associated with people in Scandinavia and the Isles, especially Scandinavia. And then our group of Cummings, Kemp and Jacobs all shared a very rare STR marker in common, DYS 447, value 21. It's very rare for anybody, actually, and of R1A people, only 1% or less, apparently, according to some surveys, have this particular marker. So two distinctive ones, one very rare, and matching on STRs in the STR test suggests this possibility here is something to explore further. So again, taking the same hypothesis states that none of these three surnames are related to each other. Ran the STR data of 67 markers through a utility known as the McGee utility to see whether that's not falsified as such, but whether there is evidence of relationship that may question that hypothesis. And again, I must stress that if you're doing genetic genealogy, you want to try and think of it in terms of what hypotheses do you have, and how can your research impact on those hypotheses, ideally by disproving them. So here we have the Kemps up here. Green shows the most probably related groups. Kemp's here, Jacobs here, showing signs of a nice strong lineage. Cummings, not really. I mean, the yellow is suggesting, yes, that all of these seem to be suggesting evidence of relationship at the 50% probability level. I've run this at 90% as well, and everything moves back. Some of the Cummings don't look related anymore, but some of them do, and yet they all still share the same marker. So we see the pattern here of fairly cohesive Kemp and Jacobs lineages, and much more diverse Cummings lineages, with a much deeper time ancestor for Kemp and Cummings, and for Cummings together and everyone together, which may be preceding the adoption of surnames, but still some evidence here of relationship. Now again, I've used another method here that's been trialled by James Irving, who's in the audience here, and it's a very good method of looking at the chance of probability of relationship. Is to use the FTDNA tip utility to estimate the probability of two people sharing a common ancestor within 24 generations. Why 24? Because that takes you back, depending on how many years to a generation, to around 1300, and again the period when surnames began to be used widely. And generally, if you're getting a 90% probability of relationship within 24 generations, that tends to be a good indicator that there is a relationship within that period. And I compared the most central Kemp against other Kemps, very high, 99% probability, against Jacobs, a bit lower, but still very high, 96, 97%, and against Cummings, even coming in around 90 here, 92, so about 90, 92%, so a bit more on the border, suggesting further back, but still implying that there is a common ancestor who may have been living around that time, around the time of the adoption of surnames, just before or around that time anyway. So again, this is utility that can draw trees, again the same sort of data. It's showing the same thing really, but again it's showing it graphically. This is root R1A down here, and up here we have the L448, which is the overall umbrella marker for these families. And down here we've got a nice cohesive Jacob lineage, consistent with their belief they have a common ancestor from about 350 years ago. Quite a nice cohesive Kemp branch as well, about the same time depth as the Jacobs one. One Kemp flying off here a bit earlier, but the Cummings again looking quite diverse. And this is a characteristic I think we're seeing of the Cummings results. They do seem to be essentially not descending from a relatively recent common ancestor. There is a common ancestor, but it could be a long way back. And I suppose there must be a possibility that it could be a coincidence that some of these Cummings branches are called Cummings. They may have actually adopted this surname quite separately and coincidentally have a similar looking genetic signature. I can't rule that out. So, what do we do? Well, we do a bit of tree building. So this is where the results we're getting from the next generation testing is coming in, and we need an overall tree to fit these results in as a context into. And this tree used to be on to quite recently on the R1A1A and all subclades project front page. They've taken it down, possibly, so they're about to do something about some of the symbols down here, which I'm not paying attention to. What I'm interested in is the SNP tree here. Here is L448. This is also quite a well-known branch, and they put Scots down here. It's not all Scottish at all. There are many people in Scandinavia and other parts of Europe in this branch, but a lot of research was done on this on Scottish people from the west of Scotland, where this was found to be quite common. And this now has a sister branch, which is this YP355 I've been talking about. This was discovered a year ago, and the people working in the group working with me have this as their own branch marker. In other words, here is this well-known branch here. This used to be a branch leading to unknown. We've now found this as their sibling marker, really, of this one. And there are many more markers we're discovering down below this. I'll show you shortly. And so this is where we're now fitting our discoveries on this strand down here, building more hierarchy in here and more depth and distinction of the tree. So let's just summarising these SNPs. So yeah, this YP355 then discovered in 2014, and this is the JAX marker. It's more people than just the JAX. There are other people in this. I've only got a couple of slides left. Very quickly, I've been working with all the people who've done big Y tests in YP355 in the past 6 months. There are 19, and 16 of them have shared their data with me. I thank them all very much. There are 2 in this room. I can't see who they are, so thank you very much for what you've done. And what you see here is modelled on Alex Williamson's big tree, though much more clumsy. It's just me typing things in Excel. It's not generated automatically or anything. We're trying to do a similar kind of thing. Link the origin point of L448, which probably originated in Norway or Sweden, with the next one down, YP355, and then we have already 5 branches have been emerging here below this, and you find the JAX in here. We've got the most distinction, if only because we've done the most tests. We've done 6 tests already, 2 Kemp, actually 5, sorry, 2 Kemp, 2 Cummings and 1 Taylor. Taylor appears to be in the Cummings brand over here, and we have 3 more in progress, so we're looking for even more distinction here in this tree. And if anybody is or thinks they may be YP355, do get in touch with me, because if you were to do a test now, you can be matched against this. You may well find that there's a potential branch that you may fit into. We may be able to find the shared SNPs that you will share with people in that branch. So in other words, you're not testing in isolation anymore, as most of these people did. There's a structure and a hierarchy we may be able to fit you into. This has led to this drop tree, again showing the same data but just showing the SNP points. One thing that I think is very interesting is we seem to have a very rapid step mutation, 1, 2, 3. These all seem to have happened very, very quickly within a couple of generations of each other. And then we have what's called a starburst here, when several branches peel off in different directions, and we know that some of these people migrated to Finland, some of them migrated to Sweden, Some migrate to the Isles. I'm not quite sure where the route is into the Isles. It may have been into Scotland, it may have been directly into Ireland, it may have been into the Low Countries and northern France, and possibly back into England with the Normans. It may have been by any other route, who knows. But those are three very possible routes that we're looking at. And what's interesting is trying to see where genealogical time comes in. And of course, for a long time, we were thinking that Kemp and Cummings, etc., would have that origin round about the dawn of surname use. But it actually looks rather not, because this is where we think genealogical time is now. And most of the branching that we identified seems to have taken place before that. There is a branch within the Cummings. We've already found a very deep branch between two strands of Cummings that seems to be going back to at least 1300 or earlier. And the branching with Kemp and probably Jacobs is also much, much earlier than that, probably actually around 500 CE, much earlier than the STRs seem to be implying. Now, how do I know that? Again, it's all smoke and mirrors, but there are techniques being developed by other people, not by me, to try and make sense, try and estimate the ages of some of these mutations. We have to be careful, there's no clock here. These are random mutations that can occur at any time in any generation. It's quite conceivable. You can get one after another in successive generations and nothing for a very long time. But the more data you get, the more you can begin to average the number of mutations at a certain time depth and get a sense of what the time depth may be. So hence, these are rather rough estimates we have of when we think these are happening. So the branching here between Kemp and Cummings looks as if it's ran a late Roman period before the supposed Viking migrations into the Isles. The JAX mutation I mentioned earlier, the DYS447 mutation, has now been found in one person in Norway, which means it may have occurred in Norway before coming to the Isles and may have occurred in this block, so it's also itself of considerable age. What I generally found in doing this is that the time depths we were becoming comfortable with through STR testing are moving backwards and becoming older. But of course, what we need is to get more tests done, as people do tests, and to fit those results onto the tree, which is giving us more of a hierarchy for here, but also for these which are developing in the same direction. There's some more big Y tests in progress for these as well at the moment. Just a very quick point of comparison, here's a tree I showed in Dublin last year, where I gave a very similar talk about this 6 months ago before this big Y data was coming in. And in October, I thought this is where the genealogical timeline would be, well before the JAX marker and the branch between Kemp and Cummings at least. And I thought these also would be much more recent, YP355, round about 0. But going back here, you see that we've got a different picture now. The time depth is stretched because of the sheer number of point mutations we're finding, and the time that's believed that these occur over generations. So, finally then, this is a little magnified view, because I know there's some Cummings in the audience who might want to see how the tree is looking. So here's a magnified view of how it's looking again with Cummings along here. And we're expecting to add a few more in here in a few weeks' time. So I'd like to end by thanking Tim Cummings, who has contributed a lot to this project, and I think will be watching from New Mexico and at some stage. I'd thank Michał Mileski and Sven Bjorndal, the administrators of the R1A project, who've been R1A project, been very, very supportive, and all of the Big Y, YP355 testers who've volunteered their data to us to help us get to this stage, and the organisers of this event, which has been a pleasure to take part in. Thank you very much for listening. I'll take any questions if you have any.
Thank you so much, John. That was fantastic. Wow, jam-packed full of information there. Any questions from the audience? I'm going to kick off with one question, and there's a lady over here. What part do STRs and 111 STR markers still play in this whole progress?
Well, we're definitely using STR markers. So one of the next things I want to do is find a way to embed these more in the tree. But I know, for example, we have a branch down here of 2 Kemps, and there's also an STR branching marker which fits with this. I don't actually know, but I do know that some people are trying to find ways to do this, and I'd like to know more about that. At the moment, what I do is I see how STR branches I know of will correlate with this, trying to fit the two together. I don't know of a rigorous way yet to build that data in.
Nigel McCarthy is using both Y-STR 111 markers and the SNP markers in his little Scottish cluster project. Group B is where I belong in that particular one, and he's estimating that the Gleasons and the McCarthy is separated somewhere around 500 to 1000 AD. We have a question down here.
By the by, how does this research relate to your Cromwell prisoners of war being transported to America, or does it?
Yeah, that's a different project. I say I'm also administrator for the Scottish Prisoners of War project. That's prisoners of the Civil War who were transportees in 1650. This is a different project. Today I was talking about two of my surname projects. But some of those people have done the Big Y, and they're also very active in their own surname projects, so it will be relevant as well.
John, James Irving. I thought I was going to have 16 questions for you. I've got none. I'm just spellbound. Well done.
Thank you.
I agree with that completely. We have to leave it there, unfortunately, because we've run out of time. But John is going to be around for the rest of the day, so do grab him. We'll be hanging around the ISOGG stand or the Family Tree DNA stand. And if you do have any questions for anybody about DNA testing, then please do ask the Family Tree DNA volunteers and the ISOGG volunteers. Thanks very much, John Cleary.